Hello, everyone, and welcome to today's webinar, Enabling a Full Spectrum of Applications and Workflows for Flow Cytometry. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Cytek Biosciences. To learn more, visit www.cytekbio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Also, if you experience any technical issues, you can utilize this Q&A box and we will assist you directly. I'd like to now welcome our speaker, Diana Bonilla Escobar, Global Scientific Director at Cytek Biosciences. Diana, you may now begin your presentation.
Thank you so much, and hello everyone. Thank you for joining us today. In this webinar, I will present new features that are available in our software. Because if you are going from a simple antibody titration to a more advanced application, we offer you streamlines and intuitive workflows that now have been expanded in the new version of the software, the SpectroFlow 3.3. So that's the main goal for this webinar. We have seen in this 5 years since the technology was introduced by Cytek, this instrumentation, the explosion of applications using the technology, so many publications, many of them in high impact journals, and within those publications, a full range of applications. Simple ones, 1 color, 2 colors, up to more than 40 colors in certain panels. What we have also seen is that those different applications and publications cover different fields. If I show you kind of the distribution of that, most of the publications in immunology, oncology, infectious disease, a lot of emphasis in drug development, prevention as well with vaccination, but a variety of things that customers can do with the instruments and a variety of needs in terms of the workflows that need to be implemented in the software. We keep getting feedback and we collect that feedback and make sure that the feedback that's represented in changes in the software. As you have seen, if you're a current customer, we keep bringing these updates to the software. And this is one of them. And the main purpose of the webinar is to describe to you what's new in the software. But in general, if we talk about the data that has been published, the data that our customers generate, our internal data, all of that, or what our main purpose is to generate high quality data. And high quality data starts with you, with the samples that you bring, how well they were collected, prepared, stained, also your reagents that need to be high quality, their stability. And from there, it goes on us, on providing an instrument that has a great optical design, detectors beautifully placed, detectors that have great performance, great sensitivity. In addition to that, giving you a set of settings, the instrument settings that you can start using as soon as you get into the instrument. That's what we call the instrument setup. So the collection of gains, laser delays, area scaling factors that put you ready to an assay with high resolution. Part of having data quality is the assay optimization. So we provide you with tools like the Cytek Cloud that allow you to do panel design in a very informed way. Also, a lot of educational tools to guide you through the optimization process of any assay. And finally, and a key point in having high data quality and in having easy results is the software. So the talk is going to be focused on our acquisition software, SpectroFlow, and how it facilitates your ability to run applications in a very streamlined way. So if now I show you the solutions that are available in the software, where we have conventional workflows, spectral workflows, and a little bit, I'm going to talk about instrument stability. And I'm sure you are surprised that now we are talking about conventional workflows. So the first part of the talk is going to be to show you why the decision on implementing those. Since we're going to be talking about conventional, the key part is to understand the basics. What's the difference between conventional and spectral? In conventional, if you want to detect PE, what you are looking for is the peak detector. You have a designated detector to capture that peak of emission. Versus a spectral, where you have multiple detectors organized in according to the wavelength, but those detectors are going to capture the full emission profile for that PE signal. Important as well that if you have an instrument that is highly sensitive and depending on your assay, you might not need all the information from all the 64 detectors in your cytometer, but you might need only the peak detector and you might need less information displayed in your acquisition software. So that's the key point, that having an instrument with high sensitivity will allow you to support multiple applications and have multiple modes depending on your assay. So if now you open your software, the newest version 3.3, you will see two modes. A conventional mode with advantages like that you have a simplified data visualization. You will not be seeing the full emission profile, the signatures. You will not be seeing the histograms for every single fluorescence detector in your unit. You will not be seeing the gains for every detector as well, just the detector that you need for. And that's going to depend again on your assay, and all of this implemented after hearing the feedback from the customers that are not doing advanced applications like big multicolor panels, but are doing only one color. GFP, all the information they need is the peak of emission for GFP, and finally a readout of positivity or negativity for that protein. Another advantage is that if you're used to using a conventional cytometer, the workflow that you will follow in this conventional mode within our instrument will allow you to keep consistency and not having to relearn things. Definitely having less options, less a simplified visualization of your data, and less clicks to go through a certain process is going to give you fast results. If now I show you some simple applications. So we will go through 5 examples. Here, just a summary. We will discuss about what if I want to just do one dye, one fluorescing protein. What if my autofluorescence is not that high? We have discussed within these 5 years a lot about the importance of characterizing autofluorescence. But if your autofluorescence is not impactful, if it doesn't compromise the resolution of your markers, then you might not need an unstained sample. One more example, it's panels where there is no overlapping dyes that is going to be easier to go on the conventional mode. But we will go through those details with real data so you can see the advantages. And on the other end, what you will have is the spectral mode, something you're used to, you understand the benefits, which are mainly the fact that you have a full signature. In some cases, that's critical because that's what allows you to distinguish two dyes that are very similar, two states of a protein that are similar, as we will see in one of the examples. It's also useful when the unstained sample gives you so much autofluorescence, and it gives you such a variety of autofluorescence because multiple subsets are present, that if you don't extract it, you will get a resolution, a compromising resolution. Also important when your sample is so valuable and you want to get as much information as possible in terms of what markers are expressed by the cells in the sample, then you need to go to a big panel, more than 40 colors. In that case, spectral mode is the way to go because it will allow you to distinguish things that are different by the signature. What we will see in this part of the talk, it's high-end applications. Applications where you have multiple colors, where you have autofluorescence that is so high or so heterogeneous that you need to extract it. And absolutely panels with highly overlapping dyes, even if it's just 2 colors. If they are overlapping, then spectral mode is the way to go. But let's then describe. For conventional workflows, what I'm going to show you is that if you have less information displayed in the software, less information in your files, that's going to help with ease of use. And that applies to assays that have low complexity. Titration, cell cycle determination, detection of soluble cytokines using beads, even small panels where you might not need compensation controls, or small panels that need compensation controls, and we will review that in a minute. But I want to show you all of that functionality in the software by actually looking at the steps that you would need to go within the software to complete certain tasks. Now you know that 2 modes are going to be available depending on your needs and your application. So first thing you would do if you want to use the system is to turn on the unit, log in into SpectroFlow. Once you log in SpectroFlow, and if you're a customer, you have seen this blue landing page that allows you to put your credentials. Once you upload a new version of the software, you will see 2 modes. full spectrum mode, and conventional mode. But once again, depending on your needs, you can proceed with. And what you will see in terms of the modules available in the software is going to be different depending on the mode. Less options, basically, in the conventional mode. SpectroFlow is organized in modules. Modules that if you click on, they will allow you to proceed with certain features and protocols. For example, if you want to run QC, you click on the QC and setup module. If you want to start acquiring your samples, you click on the acquisition module. So let's start with kind of the process you would go through if you are in front of the instrument. First thing is to run daily QC. So step number one would be click on the QC and setup module, prepare your beads, then start acquisition and nail the process to be automated. It's going to give you great information about two things. One, is your instrument performing well? And number two, the settings, the optimized settings that I described at the beginning, named Cytek assay settings, are going to be updated based on instrument performance. So at the end of this process of daily QC, you are ready for acquisition using Cytek assay setting. One more thing that it will be available is all the QC reports or the Levy-Jennings charts for you to monitor instrument performance. QC, it's done, it passed, now you're ready for sample acquisition. The SpectroFlow software has many wizards that will guide you through steps on how to complete certain things. In this case, create a new experiment. And that's highly useful because if you are not that familiar with flow cytometry, if you are not that familiar with the software, you will understand how to do things because everything is very well detailed in a wizard with steps to follow. Let's say we want to create an experiment. So you would click or go to the acquisition module. You would then, if you selected that conventional mode, now you will see a lot of signs that will indicate that you are in that particular mode. On top, conventional acquisition, your experiments are going to be named conventional experiments. From there, the process will be the same. What information you need to create a new experiment? You need to know what fluorochromes you have. You need to know what samples are going to be run and ideally organize them in groups. You need to know what markers you are detecting, CD4, CD8. And finally, details from your acquisition, like how many events I want to record. Once you define that, that information can be saved as a template. So you don't need to recreate this every time you run the same assay. You can just reuse the template. After that, that's completed, you can start the acquisition of your samples and your compensation controls if you are in this particular conventional mode. Conventional mode has a very familiar workflow if you were using Spectral before. You will run your controls, compensation controls in this case. Big difference. Now, instead of, if you look at the instrument control tab that it's displayed there, instead of looking at all the fluorescence detectors, what are all the gains, instead of needing to adjust all of them, you will go only to the fluorochromes of interest, 2, 3, or 4, depending on what you set up on your experiment. From there, click on compensation. That's going to be fully automated, the calculation. And from there, you can start running your samples. And as mentioned before, I'm going to show you examples on when this particular workflow is going to be so valuable depending on your application. Let's start with titration. We all know how critical it is to achieve optimal resolution for every reagent, antibodies, and other reagents. What you do, you basically test different concentrations of the reagents, then define what's the concentration gives you the best signal to noise ratio using whatever metric you decide to use, in this case, displaying stain index. And basically, what I want to show you on the right is 3 plots that represent different concentrations of the antibody. On top, a non-saturating concentration. You see the positives are not at the best separation from the negatives. The next one, the saturation phase or range, what we are seeing is better separation. And what we are seeing on the last one, at the bottom, what we see is the positives are really high, similar to the second option, but the negatives are now widespread. So you increase the background and that reduces your separation. So finding the optical titer, it's critical. So you have reproducible data for your staining specificity, to have signal on a scale, and also to save some money reducing antibody concentration. So acquiring and analyzing titrations in the conventional mode, it's very easy. What you will be seeing, once again, conventional acquisition on top. We highly recommend you to do acquisition in plates, also sample preparation. But once again, if you only want to titrate the CD8 PE-Dasol antibody, that's all the gain you might want to be interested in seeing and not overwhelmed by having so many histograms. You will have an automatically generated workflow to start using it. From there, you will have negatives and positives that you can gate. You can get statistics out of that to understand the medians, the spread of the population, the RSD, and then you can use the option to do batch analysis in order to then use those numbers to identify the optimal antibody concentration. This is just one example. This is a CD8 PE-Dasol titration. You will see all the histograms representing every concentration on the left, the 2 nanograms. You see that negatives and positives are closer. If you go to the other end, 1,000 nanograms, populations are very separated, but negatives are also wider. So we need to find that optimal point where the separation is the highest with keeping the background low. Because of that, we calculated stain index. You will see it in the graph at the bottom. The stain index will represent the separation, how well you are seeing your positive population. And then calculating a stain index for each dilution will allow you to define what's the optimal antibody concentration. If we now go to another example, this is where we have got a lot of feedback from customers saying that for cell cycle experiments, you don't need all the information that is provided by the spectral mode. You don't need an unstain. You don't need a compensation control. You can proceed with your analysis with the raw data. Also important that the data that is generated will be easily analyzed in a third-party software with the conventional mode. What I'm going to show you now is kind of the steps that you would follow for cell cycle evaluation. When cells are dividing, they go through phases. They start growing, they replicate their DNA, then they start the separation and to proceed into mitosis. Well, you can use dyes that are going to incorporate into the DNA, and I'm going to give you an indication on what phase the cell is based on DNA content. So first thing you would need to do is stain your cells. Then you would start acquisition. We will see an example with conventional acquisition for propylium iodide, and then batch analysis will allow you to evaluate multiple samples from the same experiment. This is just an example. It's HeLa cells. But it was useful that the conventional mode was doing automated selection of your primary detector, once again, without taking into account all the other detectors that are present, even though the information is there and you can retrieve it in case you want to do a different analysis, like spectral analysis later. Important as well in this particular case, I'm showing you on the right 3 histograms. The one on top, samples run at low flow rate, medium flow rate, and high at the bottom. What I also want to highlight is that the data quality, the tightness of those peaks, first peak G0, G1, then that plateau S phase, and then G2, GM, the other peak, they are very tight no matter how fast you're running. So data quality is clearly not impacted by the flow rate as we are seeing in this example with HeLa cells. What I want to show you next is we described so far one color assays, but this one has 2 colors. It has APC and PE. Once again, acquired in conventional mode. This assay allows you to measure cytokines that are in suspension. The way it works, and we are using a Legendplex assay, is that you have bits coated with antibodies that are going to capture those soluble cytokines in the sample, and then you use another antibody and a fluorescence system to detect or quantify how much cytokine was present in the sample. So all you would need to do at this point is follow the instructions from the manufacturer on how to do the staining, and also the instructions from the manufacturer on how to use this assay in a conventional cytometer, because that's going to apply. So it's going to be easier to implement if someone is used to using a conventional cytometer before. What you will see from this assay, once again, is 2 colors, APC and PE. APC, based on the intensity, will give you different populations of beads that are going to capture different cytokines. 5 total in this case: IL-2, IL-6, IL-10, interferon gamma, and TNF alpha. versus PE. The intensity of the PE signal will tell you how much cytokine you have with those capture beads. What we're gonna be, what's clear for this particular assay is that you don't need an AND stain. You don't need compensation controls. You could proceed with raw data in the spectral. Customers do that, but it's easier if you have the name already there. If for analysis, you already have the fluorochrome name instead of having to define what's your peak detector. Having all of that automatically assigned saves you time. Of course, if you want to know from those cytokines, what's the concentration, how many picograms per ml you have in your sample, then you want to generate a standard curve with known concentrations of the cytokine, and then identify for your samples which ones were present at which levels. This is the evaluation of those cytokines. What I'm going to show you on top is a plot for the unstimulated, on the bottom for the stimulated. So on the y-axis, you see two populations. The one on top, interferon gamma, the one below that is TNF-alpha. So you compare unstim and stimulated, you see that the intensity on the x-axis for the PE signal is as expected, lower in the unstimulated and higher upon PMA stimulation. So now, as I mentioned before, if you create a calibration curve, you can know how much cytokine you have in their concentration of the cytokine. So in this plot, what we are seeing is in blue, the unstimulated, low signal as expected once again. But upon stimulation, what we are seeing in the undilute sample, the first red dot, it's high concentration, and how that concentration decreases, proving that the system is working well once the sample is diluted. And it's not only about one or two colors, it's about also panels, multicolor panels in which the fluorochromes don't overlap. So since now we're going to talk about panel design, I'll be giving you few details about the Cytek Cloud, because it's such a useful tool that we have available as part of the software, but it's definitely an online tool. It will allow you to do smart panel design. It's going to guide you and with all the information that it's needed through a wizard for a fluorochrome assignment. So if you don't have a Cytek Cloud yet, you can go into our website, create a new account, and start using the benefits, which are fantastic. Fantastic, why? Because we in the cloud have information for a huge database of reagents. We have characterized more than 300 fluorochromes. We know their brightness. We know how much spread they introduce into each other. We know how well they would pair with a certain antigen density in your marker. And we also know how unique they are if you want to put them in a combination. So the Cytek Cloud is great to select the fluorochromes that you want to use for your panel. From there, you will get a report. You will be able to look at the fluorochrome selection in this grid format that allows you to do QC. And even better, since you put all the information that you need for markers and fluors already in the cloud, that can be translated into an experiment template ready to go whenever you go to the cytometer and start acquisition. So we wanted to show you the benefits of using conventional mode. This is a panel of 7 fluorochromes that was optimized, understanding that they have minimal spectral overlap. As I mentioned before, if you use the conventional mode, you will be focusing on the information coming from the peak detector. So peaks here are well separated. The key point of this panel was to detect TBNK, so T cells, B cells, and NK cells. Simple panel, but once again, a perfect match for that conventional mode. So if I now show you an understanding from the signatures that there is no overlap. If I now show you how the panel looks if I don't do any compensation versus if I apply it just as a proof of principle. So you are seeing 5 gates for each condition. No compensation on top, compensation applied at the bottom. A lot of populations, a lot of markers, but key point is that if you look at any of the columns, what you are seeing is that our ability to call a population, to gate it, to define its frequency, to define its intensity is exactly the same. These fluors have been selected so you don't need to use compensation controls. We have proved that it works. So it's another way to go in terms of conventional when your assay is so simple that we have proven compensation controls are not needed. And of course, if you change specificities, if you change the fluors, you might go through the validation process, but it's definitely doable. One more case, and we have heard this a lot from flow cytometry course, is the need for backbone panels. Backbone panels that can be adapted to any of the instruments that you have at the core, conventional systems, spectral systems. So we designed this 8-color backbone panel. It is for a 3-laser configuration, so a lot of space if you wanna expand it. The panel is focused on T cells. So from the panel, you can get the T cells that are TCR gamma delta, the ones that are CD4, CD8, and out of that, a little bit of information on the memory status from those T cells using CD45 array and CCR7 to get the 4 different subpopulations. Well, I'm going to show you now the 8 colors that we selected for this particular assay. If you look at the normalized spectra, you see that now there's overlap. So proceeding without compensation control is not an option. We tested it anyway, but definitely controls were needed. And in this particular assay, we use only beads because they gave us great results. Important this type of backbone panels because they can, again, they can be implemented by a customer that only needs these markers. They don't need anything else. They can just proceed with comp. But it can also be implemented by customers that want the backbone but now want to extend, as I will show you later. And definitely for that, you would go for the spectral mode. So if I show you now the panel performance, using controls, using 8 colors. Once again, many markers that you see there, many populations. What I want you to pay attention to this time, it's how well resolved every population is using conventional mode. We have done a lot of testing comparing conventional and spectral to make sure that this is a good recommendation for you. That if your peak detectors are far apart, that if you use conventional, you still can do a great quality analysis of your data. Also, I want you to pay attention to the frequency, the intensity of the signal. We see markers like TCR gamma delta, this is the second row, first plot, that is low express, low frequency of the positive population, beautifully distinguished, beautifully resolved, versus marker in the next plot out of the CD3 population, markers like CD8, CD4, highly expressed, where you expect to have good resolution. So every population, every marker, it's well resolved, and that's something critical whenever you are evaluating an assay in conventional or spectral mode. Now I'm going to show you spectral workflows. We have discussed so far applications that are less complex, applications with 1 color, with 2 colors, with few colors that don't need all the information that spectral workflows provide you. But now, since if you want to make the decision later, if I want to go conventional or spectral, then you would do it based on your data, based on your application. I want to then highlight which applications you for sure need a spectral workflow. It's basically 2 cases. First, if you have highly overlapping dyes, the way to distinguish them is not having a peak detector, it's having the full signature. The other situation is when your autofluorescence becomes problematic and you need to extract it. So we will see 5 examples. First, I'm going to show you that backbone panel that was optimized for a conventional instrument or a conventional mode in our unit. What if I want to extend it? What if your customer comes to you with more markers that they want to add to it? It's not only about more colors. It can be about difficult signatures that cannot be distinguished by the peak. Like this example that I'll show you, a fluorescent protein that is going to have a shifting spectrum based on the redox state. Then, of course, panels with highly overlapping dyes is a good example. And I'll dedicate the last part to autofluorescence. Show you samples that have high autofluorescence. Show you samples that have multiple autofluorescence, how we extract them, how the software provides you with workflows that are simple to use to do this more complicated tasks. Important to mention that unmixing is completed in a very similar workflow compared to what you would do with compensation. So you run your controls, in this case, reference controls. You will see the raw data, you will see the signature, you will be able to QC the signature and identify differences between signatures in the fluors in your panel. You will proceed with unmixing. It's 1 click in the software. All the calculation will be done automatically. Then you will run your samples and you will be able to see in your unmixed data, your markers, your fluorochromes, and the resolution of your populations. So going back to that 8-color core panel that we use for T-cell evaluation. Your customer was happy using this in the conventional, less information for them to take care of, compensation controls, simple setup. But now they have a different project. They want to go and do expansions of this panel. They now want to define T helper subsets. So they want to include a lot of chemokine receptors. The only way to go and include fluorochromes, once again, overlapping fluorochromes is the key, now is to switch to the spectral mode. In that case, if you were using conventional mode before, it's such an easy switch. To open the different mode, you can use the same data, the same templates, and start the acquisition, adding this new information to your panel, and of course, doing all the optimization. And then your customer, that takes you to a 16-color panel now. Your customer comes and says that they want more colors because they now want to focus on activation. They want to include HLA-DR, CD38. And finally, one more extension that takes us to 21 colors in this 3-laser configuration because they want to see checkpoint inhibitors, PD-1, ICOS, TIGIT. So let's see the steps and let's see the resolution, and definitely let's see the need for using a spectral mode in this particular case. These are all the markers that we have, core markers, and all the extension also included in the core markers, the viability type. What I'm going to show you now, it's a critical step whenever you want to do an extension of a backbone panel, and is that the markers in your backbone are still performing the same. So you had seen this gating strategy. What you're seeing on the first row is the core panel and all the core panel markers. So the distinction of cells that are alive, distinction of cells that are singlets, that have small scatter, that have expression of TCR gamma delta, CD8, CD4, and then the memory status. Important then that those markers look exactly the same when you go and do extensions. So I want you to focus on the last two columns, which is CD4s and CD8s, and the memory populations, which you expect to have 4 subsets, as we will see there. You see how those four subsets can still be identified successfully in terms of defining what's positive and what's negative, even though, as you see on the first plot for the CD4 and CD8, the negatives are tighter in the core panel, expected. We are adding more fluors, more spillover spreading. But at the end, the ability to see the populations is preserved, no big changes in the other markers. So now we know my initial markers are not affected. Now let's see what happened with the new markers that we added, how well resolved they are in this particular panel and context, and how important it is to have the spectral mode in this case. The first extension was for TH subsets. So a lot of chemokine receptors in here. CCR10, CCR6, CXCR5, CXCR3. So many things that allow you to define if something is a T helper 1, T helper 2, 17, 22, and different subsets. 2 more markers, you will see them. And the last plot is CD28 and 27 that allow you to go deeper in the effector memory characterization. But key point, since there's so many markers and populations, a little bit overwhelming, is that things that are low expressed, that have low frequency, can be clearly identified and separated from the background, from the negatives. We see it with the first plot, CCR10, very nice separation of the positives. And as well, really nice separation for populations that are more expressed, like CXCR3 in the TH1, TH2 GM. Key point, that no matter the panel, the goal is to have good resolution, high quality data with that extension in the backbone and in the extension. The second extension was activation. So you are seeing CD38 in the y-axis versus HLA-DR. And each plot is representing one of those memory subsets they were interested in initially in the backbone panel. So you have naive, that's the first plot, and then the memory evolution. You're seeing in the naive that beautiful profile. Well, most of the cells are 38 positive, no HLA-DR expression. And you see in the progression of memory how 38 is lost and HLA-DR is more expressed, for example, in the fourth plot, which is the early effector memories. In a similar pattern, what we are seeing with the last extension, which is all the checkpoints, showing you here PD-1 in the y-axis versus TIGIT. As expected, first plot is naive. In the naive cells, we don't see PD-1 and low levels of TIGIT, but you see that increase even in co-expression towards early effector memory, to early memory, where you can clearly see, separate what's single positive, what's double positive, and a beautiful, beautiful proof that the assay is working, going to the end where TEMRA, CD4 positive cells will not have PD-1 or TIGIT as expected from the biology. I was telling you that it's not the usefulness of spectral is not only about adding more and more colors, big panels. It can be a simple assay. This is an example from one of our customers. Thank you for allowing us to use the data. They are from the Institute Pasteur in Uruguay. What we are seeing here is the use of a biosensor. A biosensor that, depending on the redox state, is going to give you a different signature. And we will see the signatures in a minute. The system that they use is a parasite. These are trypomastigotes from Trypanosoma brucei. And what we are seeing in the slide is three signatures. The signature on top when there is no expression of the biosensor protein. So very low background. But on the next two plots, what we are seeing is what if there is a reduced environment or an oxidized environment using GFP as the readout, as the reporter. So the key point that I want you to pay attention to is that green rectangle that it's indicating the peak detector for GFP. If we let that guide for the peak detector, there's no differences here. So conventional mode is not the best way to proceed. But if I look at the full signature, now if I look at the violet module, where you see detectors violet 5, 6, 7, the intensity of the signal in the oxidized form, it's higher. And if we have tools in the software to compare them more than by visually inspecting the plots. We have something called similarity index that allows us to compare those two. And if I calculate that number as 0.1, 1 is similar, the same, and 0, it's low similarity. So they are similar as we can tell from the plots, but also that value indicates to me that they can be distinguished as unique and they can be a mix and the data can be clear. If I show you the normalized signatures where both independently of the intensity are scaled the same, we can clearly see how they are unique and how, not using the peak detector, but the full signature, I can distinguish them and then start quantifying. I'm showing you raw data, which means peak detector plotted for the oxidized and the reduced. What we are seeing, and I want you to focus on treatment 3, because all the other treatments are basically controls of oxidation and reduction. But in treatment 3, this customer expected to have both forms, oxidize and reduce. If you use conventional or if you use just the raw data, it is going to be so difficult to distinguish them and separate them. You still can see two populations there, but quantifying them, it's difficult. Versus if you have the signature, because now the signature will allow you to distinguish them as unique, and you were seeing treatment 3 at the bottom, how two populations separate well, indicating reduction and oxidation, that redox state, which that biosensor protein, it's indicated in this trypomastigotes. So highly useful in this case where you only have the same protein with two different states, basically. The last part is autofluorescence. It's a topic that I'm highly interested in. In these 5 years, we have produced so much information about autofluorescence, looking at customer data, generating internal data, reviewing workflows, making sure the workflows are efficient, and finally implementing those workflows in our acquisition software to simplify the way you do analysis of the data. The use of our technology, full spectrum profiling, is so critical if you need to characterize and extract autofluorescence. And that comes because cells, when they pass through the instrument, they emit fluorescence, even if they have not been stained with anything. They can be, the components within the cells can be excited by the laser and then emit fluorescent. And that happens because cells have structural proteins, cellular organelles, metabolites that emit fluorescence. And important to mention that autofluorescence, the levels of autofluorescence and the pattern in terms of wavelengths is going to depend based on how big your cells are, their granularity, the type of cell, their metabolic state, or even experimental conditions, if those cells were stimulated, if those cells went to fixation and permeabilization. That's going to change how your unstained sample will look, which is the indication of autofluorescence. So let's go to the step-by-step on what to do if you have autofluorescence. And first, how to identify if you need to extract it. And this is going to depend on customer needs. That's why we have multiple workflows and multiple modes for you to decide which one fits better your data. First step, if you want to evaluate autofluorescence, you want to acquire unstained cells. Highly recommend you to record enough events and make sure that your unstained matches your multicolor samples. You have the same cells, stimulated the same, and stained through the same steps, of course, not adding the antibody, but everything else, washes, buffers, incubation times are the same that in your multicolor sample. From there, you select from what you're seeing in your signature, you select the most appropriate workflow based on the autofluorescence. And we will go next on how to select the most appropriate workflow. Once you select the workflow, you start acquiring your multicolor samples while doing the autofluorescence extraction embedded within the unmixing process. So how you don't know what's the best workflow? If you don't know how your sample looks, if you have not seen our webinars where we describe some of these characteristics, well, you run your unstain. Let's say your unstain looks like this. You are using stem cells, like in this case. You look at the signature, you see the ups and downs. You see that there is emission in the UV, in the violet, or in the blue detectors, a little bit on the others, but you can see the pattern, which is what we call the spectrum, the ups and downs. In this case, if you look at only one detector, the data is not that spread. It's condensed, indicating the homogeneity within the sample and within the cells in that sample. So if all the cells have a similar pattern in brightness, in spectrum, then you will go for a certain workflow. What's some examples? Cell lines, PBMCs, cells that have been sorted or separated, that are very homogeneous. You can also have heterogeneous samples where you not only have one population, you have multiple populations, and those multiple populations behave differently in terms of autofluorescence. Their signatures are different. This example, it's a mouse liver. You're seeing on top, the scatter plot tells you that there's multiple things in there, but the signature plot is showing you that heterogeneity. Variations in intensity. Some are low, some are high. You have multiple signatures in there. To give you some examples, that happens with bone marrow, that happens with solid tissues, brain, liver, spleen. That happens with co-cultures that involve cells that are different in autofluorescence, lymphocytes and macrophages, lymphocytes and tumor cells. So there is a separate workflow that we will describe later for heterogeneous. And another situation you could have is having a mixed experiment, an experiment where some of your samples will have a different autofluorescence because of the way the experiment was designed. For example, you have unstimulated cells and stimulated ones. You have cells that were stained fresh, cells that were stained fixed, cells that had to go through permeabilization. You have multiple tissues, blood, liver, lymph node, things that are so different in terms of autofluorescence that you need to separately extract the autofluorescence per group. So now that I show you how the signatures will look, now that I show you what are examples we have, let's see what are the workflows that are available in the software to simplify the way you do the extraction. And after that, we will see why doing extraction. Why it's mainly because you are going to improve resolution in your data and to highlight that not always is needed. So you not always need to go for a spectral mode. Back to that classification, you have homogeneous samples that are very tight, heterogeneous samples that have multiple signatures, and mixed experiments where each group within your experiment has a unique autofluorescence profile. If you have a homogeneous sample, you will straight click on unmix with or without extraction. You can do both, so you can compare if indeed your resolution is improving. If you have a heterogeneous, more complex sample, you will launch something that we call the Autofluorescence Explorer. The Autofluorescence Explorer will allow you to identify the different subsets that are there in terms of autofluorescence and then incorporate them into the unmixing for a separate extraction per population. And finally, if you have a mixed experiment, we allow you to create specific unstain group controls per group so the extraction can be done separately. And now examples. And this time we have so many webinars in case you are interested in getting more information about autofluorescence. Everything is in our website. But we tend to show big panels where the autofluorescence is so impactful that you lose your resolution for many markers. But this time I want to show you simple examples because the power of extracting autofluorescence comes also if you have only one color. And we will see that. 1 or 3 colors like this example. But let's go through that by showing you a homogeneous sample. And let's say you are very used to run fresh PBMCs. What you're seeing there is data from unstained PBMCs. You see in the scatter plot the lymphocytes gated, and then the signature for those lymphocytes, very mild autofluorescence, a little bit out of the UV detectors, violet and early blue detectors. But let's say that now you decide to define your assay, simple assay. You want to measure viability, and out of viable cells, CD3, FOXP3, and CD4. Now that FOXP3 is added, you need to change the way the cells are stained. You need to permeabilize them to access the FOXP3 intracellular target. And if I now show you how unstained cells that went through that process of fixation and permeabilization look from the scatter plot, you see how different things are now. And from the signature, now we see how the autofluorescence is higher in intensity, has a different pattern. We see now emission out of most of the blue detectors, out of the yellow-green, and even a little bit more out of the red. So things are different. You want that matching and stain. And let's see why extracting versus not extracting will make a difference. I want to point out that when you have autofluorescence that high, that's like another fluorochrome in the assay. That's adding spread, that's also adding background that will impact your ability to see some of the markers, as we see here clearly, that are going to be highlighting here the peak detector and how much light, because of autofluorescence, you are getting as background for that particular detector. So what the software is going to do when you extract autofluorescence is including that signature that you are nicely getting from your unstained sample into the calculation for the unmixing. Now I'm going to show you the data. What you see is two plots. On the x-axis, CD3 plotted versus CD4 and FOXP3. If I don't do any extraction, you see on the first plot, three populations, one that is negative and one that is single positive and on top double positive. They're so close to each other. You're losing resolution, which is, again, that separation between populations. We still can see the positives, especially looking at the other plot, FOXP3, because of the resolution that it's high in this instrument, the high sensitivity of the detectors, but it's making difficult for you to call what's positive, what's negative. If I do extraction, and this is just indicating the stain index, so you can compare. If I do extraction, now we can clearly see the separation, right? Negatives rounded closer to zero, positives better separated from that, and an easy way to define what's positive or what's negative. Also thinking that if you acquire more events, then the spread of this data, it's going to be also impacted. So useful with three colors, where you extract and you get better resolution. And the only way to extract it is if you have an unstain that has been characterized through the spectral mode. One more example, and this time to show you a heterogeneous sample that has been acquired in the spectral mode, and then we have used the Cytek Autofluorescence Explorer to understand the heterogeneity in there. These are mouse macrophages. From the scatter plot, you can tell it's all over the place, right? Low complexity, high complexity in the cells. And if we look at the signature, it's the same. For every single detector, you have intensity values that go from low to high. And even if you look at the full signature, especially looking at the last part, the red area, we see like two things there. So multiple populations with different signatures, with intensities all over, that need to be distinguished and whose autofluorescence need to be extracted separately. What we are seeing next is the, just to give you the, back to that example, this is how a homogeneous data looks. Very tight CVs for each detector versus all over, or in terms of high spread in the data. In this case, only one color. All we needed was to define viability in these macrophages upon treatment by using DAPI. But if I show you the autofluorescence, that's overwhelming DAPI. You might not be able to see it just because your autofluorescence is so high. What steps you would follow? First, Autofluorescence Explorer. It's available with right-click on your unstained sample. From there, second step is to identify what signatures are there. You will do it based on intensity, based on the critical detectors. Those signatures, once identified, the software will automatically organize them, normalize them, provide you with metrics, because it's not only having them identified, it's having a way to understand if they are unique, if they are different or not, based on brightness, based on uniqueness. So, and the spectrum as well. In this particular case, two of the ones that were identified were similar. So they would count as one, like one fluorochrome. and one more that is very different. So 2 extra fluorochromes in your assay. So those will be automatically created, a file for each of them, and they will be part of your unmixing when you proceed with unmixing with extraction. Next is now look at the data and look at the benefits of doing that, of understanding that that sample had 2 unique signatures that you need to extract them. And let's now look at the data. This is how DAPI look. So if you don't run any unstain, if you don't go through the spectral mode, you would conclude from here that half of your cells are dying. But if I show you the unstain, plotting DAPI, but no adding the dye in there, it looks exactly the same. Very few differences. The autofluorescence was creating all those false positives. So you need to find a way to understand it, running the unstained sample, and then to extract it using the workflows, automated workflows in the software. Once you extract it, now I'm showing you unstained in the first plot. There's no signal if the unstained is displayed versus the signal that is shown for DAPI in the stained one. So now we can clearly see how different it is in terms of data interpretation when you go without extraction, without an unstain, and using the proper unstain that will allow you to quantify viability in a much more effective way. The last example, it's having multiple groups. In this experiment, we had cells that were stained, only surface staining, cells that were stained for intracellular targets, and within those intracellular stainings, cells that were stimulated and unstimulated. So that's 3 groups. For each group, you have the ability in the software to add an unstained sample that will allow you to do specific extraction depending on what you have in your unstained sample. You look at the signatures, they look very different. Now we look, simple panel, CD3, CD4, CD8. If you look in general, comparing across the 3 plots, the patterns are different, it's different stainings. The patterns are different also depending on the stimulation. But now having a specific unstained control per group will allow you to extract separately. So as you see, all the negatives pointed out by the arrows are around 0 with good symmetry around 0, and then you can really see the separation for your positive populations. So that's the end of the spectral part. Last part that I wanted to show you in this last 5 minutes, it's how some of the tools and features that are in the spectral flow software will allow you or facilitate you to do longitudinal and multi-center studies. The high stability and the ability to use optimized settings facilitate things like running samples across batches in the same instrument across different days, or across instruments in different institutions, or across different platforms, like you have an analyzer and you also have a sorter, you want to transfer the assay, how easy that's going to be because of the instrument stability. And because of something I mentioned already, but I want to add a little bit more of background, is the Cytek assay settings. It's our approach that we have demonstrated that is very robust for most of the applications, and it's an approach to instrument settings. All instrument settings consider all the values that you need to start acquisition, the gains for your detectors, laser delays, area scaling factors, all of that, it's ready to go. How we did it, we established those settings based on biological samples, how they perform. And important that every time you run QC, those values are going to be automatically updated based on instrument performance to give you a consistency across time. That's great for ease of use. You need to just click on Cytek assay settings and start acquisition. It's great for reproducibility, as we will see across instruments, days, and platforms. And finally, that's what provides stability over time in your measurements because of the update based on the daily QC and instrument performance. First, showing you across days. This is a study, a COVID study by one of our collaborators at the German Institute in Barcelona. They use a 37 color panel that was run on different days, reusing controls. But key message is that 1, you can reuse controls, and 2, for the same sample across days, you get similar results, and important that that can be done. Similarly, in this comparison that we did across 20 units, this is an internal comparison using units from customers all over the country. This is using an 18 color panel, lyophilized cells that were pre-stained and then run on the different instruments to see the consistency. 20 units total. We have a webinar that described in detail the results. Here just showing you 3 instruments to facilitate the visuals. For every column, just pick a column, you will see how consistent when you run using Cytek assay settings, the same sample in different instruments that were not harmonized, like taking into account fluorochromes, just using Cytek assay settings, which is a very robust approach, we get similar results. Similarity in what? In where the gates are placed, in the intensities, in the frequency. So it's a good starting point if you wanna start harmonizing in units. And the final example, and just to summarize everything, is that if you're using an Aurora system and you decide that some of the cells that you characterize, you want to go deeper analysis in their functionality, for example, so you need to sort them, then you can match that with an Aurora sorter system. In this case, showing you data using a 25 color kit that was designed by Cytek and is available for you to purchase. And it's the what we are seeing next is the results. Aurora on top, sorter on the bottom, multiple populations identified, but the key point is that now transferring assays from an analyzer to our sorter is simple if they have the same optical configuration and if the instrument setup has been harmonized by using Cytek assay settings. We see every population, like if we focus on the first one, TCR gamma delta, beautifully shown, and we can tell how the separation is great no matter what instrument is being used and what platform. So now to conclude, we I hopefully have demonstrated to you that it's not only the hardware, it's not only the instrument, but also the software and how well it is integrated to offer you full spectrum of applications or workflows. It's having us in automated setup, it's having simple workflow, it's having solutions depending on what samples you're bringing, and it's giving you flexibility in fluorochrome selection with that optical performance. It's benefiting, as you saw, different applications. It's also allowing you now to use conventional or spectral modes based on your needs. If you only need one color, conventional. If you need overlapping dyes, autofluorescent extraction, spectral. You saw how data resolution can be so empowered by extraction of autofluorescence. And finally, I want to say thank you. Thank you to the fantastic scientific commercialization team at Cytek. Thank you to our customers for providing feedback, for letting us look at your data, and that way letting us improve in all these workflows and in the way we teach spectral cytometry. Thank you so much, and I'll be happy to answer any questions.
Thank you, Diana, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question says, how can you handle autofluorescence in the conventional mode?
So back to that idea that if you want to do autofluorescence, characterization and extraction, the way to go would be having the full spectrum signature. So we highly recommend that if you identify that your sample has this diverse profile of autofluorescence, or if your sample, last week we saw, has high autofluorescence, even if it's just one population, then go for spectral, look at the end stain, look at the profile, look at the diversity of populations in there, and then decide based on the workflows that I explained which one you would go for, but spectral would be the best way to go.
Excellent, thank you. Our next question here says, do you need to rerun QC if you go from one mode to another?
That's a good point. No, you don't need to redo the QC. You will be, let's say you start on conventional mode, your customer runs the QC or the user runs the QC, then that QC information will apply for all the customers on that day, independently of the mode that they decide to use.
Wonderful. All right, our next question here asks, can I reuse compensation controls on conventional mode?
Yeah, the system will work the same, right? The controls will be stored in your experiment. The adjustment for those compensations in terms of instrument per daily instrument performance will be done. So you have the ability to reuse them, understanding that all the pre-analytical phase need to be preserved in terms of using the same reagents, using the same protocol, and exposing those reagents to the same steps in your staining protocol, as well as the way the samples are prepped and preserved.
Phenomenal. Thank you. And it looks like we have time for one more question. That question asks how to distinguish data acquired under each mode.
Yeah, there's going to be multiple indications in your C drive. You will see of folders, separate folders. If you export the experiment, the folder is going to be named conventional instead of raw or instead of a mix. Another one thing that you could do is decide that you don't want raw data anymore. You only need the conventional data. That's one thing. The way in which the experiments are going to be named will also have an indication, comp, at the end. So multiple ways you will realize the particular mode was used, as well as in the file properties whenever you have a file display on SpectroFlow.
Wonderful. Thank you again, Deanna, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Cytek Biosciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.